Students, in human health and disease, we have studied introduction and diseases. Next topic we have to study is the immunity. What is immunity? What is its importance that we are going to study today? Every day, when we are exposed to a large number of infectious agents, the body is being protected from that harmful pathogens by immune system. That means when we expose to microorganisms, our body is able to identify them first. Then they will be eliminated or destroyed easily so that our body is being protected. So this entire process happens daily so that the body is being protected. And consider all exposures are not leading to diseases. Only a few exposures may lead to diseases that we may face in our day-to-day life. Now, if you consider what is this immune system or what is our defense mechanism present inside our body, that we have to know. For that, the network of organs, cells, proteins that protect the body from harmful infectious agents such as bacteria, viruses, animal parasites, fungi, and even tumor cells is called as the immune system. Means it is a network of cells, network of organs, and even network of proteins also, which act together to kill or destroy that microorganisms or germs are together called as an immune system actually. Now if you consider the immune system have this ability, what is that ability? To eliminate disease causing germs, that we are calling as an immunity. So observe the definition now. The ability of the host to fight the disease causing organisms conferred by the immune system is called as immunity. Right. Now, what is immunology? The branch of biology which deals with the immunity or immune system is immunology. Edward Jenner is known as father of immunology. The immune system is working with the primary functions like distinguishing between self and non-self. This is very important function. Why? Because it has to identify its own cell and even it has to identify the foreign cell or non-self cells also. That means if any bacteria is entered, it will identify that one as a non-self which is not belongs to us. So an army of cells or army of proteins are going to be released at the time. So they will identify and next consequence are going to happen there. So this is the primary function what we are going to discuss here. Coming to the second property, that is the protection of the body. When it is identified, when it is eliminated from the body, automatically our body is being protected. So it is very important system in our body in the defense mechanisms. Right. The immunity is now divided into two different types: innate immunity and acquired immunity. What are the significant differences we are going to discuss in our coming classes. But now the innate immunity we are going to study right now. What is an innate immunity? The innate immunity is a non-specific type of immunity. Why it is a non-specific type? Let us observe carefully. Specific means it is acting particularly on a particular organism is called as a specificity. But here non-specific we are calling means if it is the microbe may be a plant, a pathogen, it may be useful, or it may be harmful bacteria, virus, fungi, like a number of germs are existing in the environment. Whatever it is going to enter into our body that are non-specifically, non-targetedly, they are going to be eliminated. That's why we are calling it a non-specific. Consider it is going to identify the self and non-self first of all. Okay, and that too it is a non-specifically. That's why we are calling it a non-specific immunity. Second property, present at the time of birth. It is an immunity which is existing starting from our birth, and so long it is going to protect us. Next, provides different barriers which prevent entry of foreign agents. So it is going to accomplish a different type of barriers which are going to eliminate easily all type of foreign agents. So these are the different properties of the innate immunity. There is a different type of barriers we are going to observe in the innate immunity, because it is going to give or accomplished by different type of barriers to prevent the entry of foreign agents. Actually, we are going to divide them into first line of defense, second line of defense, and third line of defense also. Right now we are going to study about the first line, second line, the third line we are going to study under the acquired immunity. Right. So what is that first line? Suppose if any microbe is entering for the first time or from outside to inside, then it has to traverse or it has to reach out this first line defense. If it is escape successfully, then it will enter into the second line that we are going to study now. Okay. So there is a number of barriers, four barriers totally we are going to study. They are physical barriers, physiological barriers, cellular barriers, and cytokine barriers. A physical barrier which prevent the entry of microbes. Carefully also, it is a physical barrier, means physically it is forming one obstacle for the entry of microorganisms, so they can be prevented. Right, examples are skin and mucus coating. We know skin is the largest organ present in our body, which is covering complete our human body. So it is acting as a main barrier in the first line of defense. So it is a main barrier, and if you observe, the skin is made up of many layers, keratinized stratified epithelium which is present along with the toughest layer called stratum corneum, which prevents especially the entry of microorganism. If you observe here, I am showing a number of layers of the skin, and it is also covered with the hair follicle and hair. So if any microbe is entering, it is being protected, it is going to be prevented its entry. Right. This about the skin. Next, mucus coating. A mucus is a sticky substance, you know. Then where it is secreted from? Right. So the epithelium lining of our respiratory, gastrointestinal, and urinogenital tracts are covered with the mucous membrane. A mucous membrane always secrete mucus. So what is the function of mucus actually? It is going to trap the microorganisms because it is a sticky in nature. So obviously it will trap if any microorganism is binded over here. So automatically it will be stop its entry. So unable to enter into the respiratory, gastrointestinal, whatever it may be. So it is being protected. So these are the physical barriers what we are going to study, right? Coming to the second part, that is the physiological barriers. Physiological barriers. Carefully observe. What is the physiology? The term related to metabolism, what we know. So it is obviously important for the growth also. So it is the physiological barriers are the things which are helpful for us, and even it is preventing the growth of bacteria also. How it is acting? Let us observe. The physiological barriers are many types. In that, acid in stomach. We know acid, it is because of the concentric HCl secreted in the stomach. And this acid, importantly, useful for the activation of some enzymes. That is the physiological function. And one more function, the same HCl can act on the bacteria so that it will be killed. Suppose if any bacteria or any pathogen is entered into our body through the food, it will be digested in the stomach only. So that is the first function. Next, saliva. Saliva from the mouth. We know it is containing amylase, important for the digestion. But one more function is existing. There is lysozyme enzyme. The lysozyme enzyme will act on the bacterial cell wall so that the bacteria will be killed. This is another function, right? Next, coming to the tears from eyes. Tears also contain enzyme called lysozyme. Same, it is going to prevent the bacteria. Then coming to the body temperature. Body temperature is suitable for the metabolic roles, we know already. But whenever any microorganism is entered to stop the growth of that microbe, our body or our immune cells are releasing some pyrogens. A pyrogen is a chemical which will increase the body temperature. So in fever what happens? Definitely the body temperature rises, which is not suitable for the growth of microorganism. So the body temperature which is not suitable for the growth definitely kills that germs. Coming to the sweat. Sweat which is secreted from the sweat glands is containing low pH or acidic pH, that is 3 to 5. So it may be around 3 or 5. So what happened? Definitely if any microbe is attached to that sweat, definitely they will be killed. Coming to the sebum. Sebum is oily secretion from the skin. Again, it is a sticky in nature, which is not useful for the growth of microbes. Right? What we have studied in the physiological barriers, it is not only useful for us, and even it can kill that bacteria or germs also. That's why we are calling physiological barriers. Means two different functions they are having. Right? Coming to the third one, cellular barriers. Okay, now we just observe the physical barriers and physiological barriers. Both will come under the first line of defense. Means from the environment, if any germ how to encounter our body or how to enter into our body, it has to reach these two and it has to escape from these two barriers. Suppose if these barriers, if they successfully escaped, then they will enter or they will be encountered by these two barriers. Let us observe these two cellular barriers and cytokine barriers, which we are studying under the second line of defense. Right. So, what is a cellular barrier? The name itself indicates different kind of cells are existing that to immune cells. Okay, so these cells are acting like a barriers. What they will do? They will destroy the microorganisms. Okay, let us observe a different type of cells existing in the cellular barriers. The WBC, which is going to be a neutrophil. There is a different kind of WBCs are existing in our body. So one of the type is neutrophils. We can call them as a polymorphonuclear leukocyte. What is a polymorphonuclear leukocyte? Why we have to call this one? Because the nucleus is having different shapes. Okay, and even different lobes also existing. So polymorphonuclear leukocyte it is. And did you observe there is a cytoplasm with the granules? We know the WBC is having two categories of the cells. One is going to be granulocytes, one is going to be agranulocytes. Right. So it is a granulocyte type like basophils, acidophils, neutrophils. So these three will consider, but our topic is running the neutrophils actually. So neutrophils containing this granulocyte, okay, along with the polymorphonuclear also. So this is going to be neutrophil. Coming to the next part, that is the monocytes. A monocyte is the largest WBC type of cell. Okay, it is the largest WBC type of cell, and there is single lobe, and the cytoplasm is also not having any granules over there. Okay, so this about the monocyte. What is the function of monocyte? A monocyte normally roam in the blood vessel, peripheral blood vessel. Suppose if any microbe is encountered, then it will reach to that site through the capillaries, through that tissue. Okay, after entering into that tissue, it is converted into macrophage. There is a different kind of macrophages, okay, that we will study. Okay, coming to the natural killer cells, in short we can call NK cells, natural killer cells. It is one of the type of lymphocyte. There is a different kind of lymphocytes again. B cells, T cells, etc. So one of the lymphocyte type is natural killer cells. These natural killer cells will secrete perforins on encountering any cells, especially infected cells. Okay, suppose if it is an infected cell, the NK cells will release the perforins. Sorry, these perforins are binded to the cell surface, so they will form some pores. Because of the formation of pores, definitely the cytoplasm will ooze out and the cell will be lysed. This is a mechanism existing in the natural killer cells. Okay, coming to the tissue, different kind of tissues are having different type of macrophages. We already studied that monocyte, after encountering of any infected cells or any bacteria, so it will reach that tissues and convert it into macrophages. Macrophages like Kupffer cells in liver, microglia in nervous system like brain. Okay, and in the same way osteoblasts, like there is a number of cells are existing in our body in different tissues, right? So if any bacteria is entering into the liver, okay, supposed to be, then the Kupffer cells existing over there will act on that bacteria by a mechanism, phagocytosis, and destroy. So the different type of WBCs and the tissue macrophages all work Okay, and they will do the phagocytosis process. Okay, so what is a phagocytosis? Carefully we are going to observe now. Right, the macrophage I am showing with the M here, and the bacteria is going to be indicated with B. After encountering that, phagocytes will move to that site, okay, and they will form a pseudopodia. Okay, so this is a pseudopodia, and even it is looking like the amoeba somewhat. Okay, so what happened? It will engulf. First mechanism is the engulf, means they will trap that one. So definitely there is a formation of a vesicle like structure or bag like structure over that bacteria. And consider it is a lysosome. What is the function of lysosome? A lysosome is containing digestive enzymes, acidic enzymes, and different type of proteins also. But here the acidic enzymes and lytic enzymes are going to be act on the bacteria, so it is destroyed into pieces. Okay, so what we have observed, it was engulfed. After engulfing, forming of a phagolysosome. After that, formation of particles or digestion. So this complete process we are going to call as a phagocytosis. Phagocytosis. So this is the mechanism behind the cellular barriers. Right. Coming to the fourth one and last one, that is the cytokine barriers. Cytokine. First of all, what is the nature actually? Cytokine is a type of protein secreted from different kind of immune cells. And if you consider, proteins mediate interactions among cells of the immune system. So our immune cells will secrete the cytokine. These cytokines will do the cellular interactions. Cellular interactions means they will communicate with each other and take the necessary step. Okay, so in the immune system actually, that is the important thing what we have to study. Coming to here, there is a different kind of cytokines involving in our body. Only two we are going to study here, that is an interferons and interleukins. Interferons, it is a protein secreted from virus infected cell. Okay, and it is going to protect the non-infected cells from further viral infection. Okay, so in this diagram I want to show you a small process that is happening inside our immune system. Okay, consider it is a virus infected cell. The virus infected cell already having number of viruses. Okay, so they are ready to move out from that cell and they can destroy the neighboring cells actually. So before to that infection only, here the virus infected cell is secreting some red color particles. You might be observing. Okay, these red color particles we are calling as a cytokines or interferons. Okay, there is a different type of interferons, interferon alpha, gamma, like that. So there is a different types over there. Okay, we are not concentrating now, but the interferons are going to be communicate and they are going to be bind to the neighboring cells. Okay, so they will bind to the surface receptors. Okay, so after receiving that signal, the cell is now protected. Means even though the viruses are present here, they are unable to enter inside. They are unable to infect that cells. So the cells are being protected in this way. Clear all of you? So this is the mechanism in the interferons action. Coming to the interleukins. From our leukocytes, there is a protein component which is going to be cause growth and differentiation of different immune cells. Okay, so we know already the growth is nothing but cell division and the differentiation is different formation of like mature cells from the immature cells. Okay, so this is a differentiation that we are going to study in our next few topics. Okay, so if you have any doubts regarding this part, you can keep the comments portion. Okay, thank you all of you.